finished up and then I didn't start. Going to the internal structure of the muscle cells. In the myofibrils, we have a repeating pattern called a sarcomere. This is also referred to as the functional unit of muscle because it's the smallest piece you can go down to that can still do the muscle's job, which is contract. It's a particular pattern of myofilaments. The filaments are thick and thin. They alternate as you go along the myofibril with some overlap between them. The edge of a sarcomere is called a Z-line. The thin filaments are attached to the Z-lines on both sides. And extends toward the middle of the sarcomere. The thick filaments are lined up in the center of the sarcomere. That area is called the A-band. Within the A-band, there's a stretch where you have just thick filaments and no thin, and that's called the H-zone. And then down the center is the M-line, M for middle, and that is some proteins that hold the thick filaments together, so they stay in that spot. There are also thin filaments sticking off the other side of the Z-line into the next sarcomere. The portion of both sarcomeres where you have thin filaments but no thick is the I-band. I-bands are the light ones. They have just the thin filaments. A-bands are the dark ones. They have thick filaments, including the portion of overlap. So on here, this bracket indicates one sarcomere. These are the Z-lines. You can think Z for zigzag. I don't remember where the term actually comes from. These are the thick filaments, so everything here is an A-band, and then where you've got just thick is H-zone. And these dots indicate where the M-line would be connecting the thick filaments. So then from here to here would be an I-band. Okay, on structure of a sarcomere. The filaments themselves are made of proteins. The thin filaments have 3 different proteins in them: actin, tropomyosin, and troponin. In a closer view, actin looks like kind of a string of pearls. Actually 2 strings together. That takes longer to draw. And actin is, it's named for active, even though it's not doing anything, it's being acted on. It has sites on it that the thick filaments bind to. So it's kind of like a road where thick filaments can pull and travel along it. Tropomyosin is a roadblock. It wraps around the actin and it blocks those binding sites. That prevents your muscles from contracting when you don't want them to. When you do want them to, you need to get the tropomyosin out of the way, and that's what our third protein is for. Troponin is attached to the tropomyosin at intervals, and it can pull the tropomyosin out of the binding sites on the actin. That allows the thick filaments to attach and the muscle to contract. So that's your thin filaments, 3 proteins. Your thick only have 1 protein in them, which is called myosin. A piece of myosin by itself looks something like this. It has a tail and a head. When you put them together in the thick filaments, their tails are stuck together, and the heads are sticking out all the way around at both ends. The myosin is what's actually going to do the contracting. It's going to pull on the actin. How that works, we'll come back to. This shows myosin here, actin in the lavender, tropomyosin in kind of bluish, and then these side clusters are troponin. Any questions on filament structure? Moving back out to look at the cell, I need to erase this sarcomere to have some room. It's a cell, so it's got a cell membrane. We're just kind of cutting it off at one spot here so that we can see all the parts. The cell membrane is called a sarcolemma. Sarco means flesh, and it's another root that gets used with muscles. So we have myo, which is muscle, sarco, which is flesh, as we saw also with sarcomere. The cytoplasm is called sarcoplasm. You have the myofibrils that we already mentioned. Sarcoplasmic reticulum. Here's another, we take the word for cells in general, cut off a piece and replace it with sarco. So instead of endoplasmic reticulum, we have sarcoplasmic reticulum. Specifically, that replaces the smooth ER, so it doesn't have ribosomes. It has a special arrangement where it wraps around myofibrils in kind of a net. And then there are several rows of it. Down the length of the muscle. It has an additional job that's important in muscle cells, which is storing calcium. The amount of calcium loose in the sarcoplasm is going to affect whether the muscle can contract. And then there are indentations of sarcolemma called t-tubules. The T stands for transverse because they go across the width of the cell rather than lengthwise. They also wrap around the myofibrils. There will be another set here. So they come through in between all the sets of sarcoplasmic reticulum. Okay. So you have alternating T-tubule sarcoplasmic reticulum along the myofibrils. From the surface, it's kind of like if you had a log of Play-Doh and you jabbed your finger in at a bunch of places. So it's just little holes going in. But then on the inside, they branch out and wrap around like this. So here we've got sarcolemma, openings of T-tubules, and then this shows them going in. Orange is the sarcoplasmic reticulum. These are your myofibrils. The blue over there is a nucleus, and all these little yellow things are mitochondria. So they still have the standard organelles. In fact, you might recall from the tissues chapter, muscle cells have a lot of nuclei along the length of the cell. They also have a lot of mitochondria because it takes a lot of energy to contract your muscles. Questions on cell structure? All right. So what did we say muscle tissues can do that no other type can? Yeah, they contract. That means they get shorter than their resting length. Now, if you have a muscle that's anchored on both ends to bones, and you shorten it, what happens? Yeah, it pulls on those bones. So let's say my biceps here, it's attached at my shoulder and just past my elbow. If it shortens, it pulls on the two ends. This end is more anchored because it's against my trunk, and so my forearm is what moves. So that's the wide-angle view of how muscles make your body move. This animation looks at that and introduces what's going on inside the cells.
We can stand, sit, and move because of the musculoskeletal system. 206 bones, 600 muscles, and more than 1,200 joints. Muscle movement requires the cooperation of both the nervous system and the musculoskeletal system. Movement occurs when groups of muscle cells contract. Electrochemical impulses from the nervous system stimulate fibers called sarcomeres, causing them to shorten. Contraction continues until the impulses cease, and the muscle once again relaxes. As muscles contract, the bones to which they are attached move. Joints, lubricating structures between bones, make movement smoother. Thanks to the teamwork of muscles, bones, and joints, locomotion is possible.
So for the muscle to contract on the wide view, or the organ level of organization, the individual cells within it also have to get shorter. The way the cells shorten themselves is called the sliding filament model. It's kind of like a game of tug of war between the myofilaments, where the thick filaments are the players, the thin filaments are the rope. The thick will pull on it, and the two teams get closer together. The whole sarcomere shortens. And because the sarcomeres are linked together at the Z lines, and you have multiple sarcomeres shortening throughout the muscle, it's going to pull the whole myofibril shorter, therefore the whole cell. Here we've got Z lines and thin filaments in purple, thick filaments in pink. They pull the thin together until they bump. That's as far as it can shorten. So this is going to look at the sarcomeres, and then we're going to come back and look at the filaments and the steps they go through to make this process.
Filaments lie side by side, and the H zones and I
Lost some of the sound there. So this is going to tell us a little about the filaments and also